Hi. Today we are going to extend our knowledge of genetics by taking a more nuanced view of Mendelian principles and extending them to explain interactions between alleles of the same gene, between different genes, and also explore the influences of the parental genome on the progeny's gene expression. Please prepare some questions for question time as always, and here comes the voiceover. These are some mosquitoes that I study in my lab. In fact, we made them. Well, at least we genetically modified them. And what we were trying to do was alter the pigment in their eyes. And if you look closely, you can see three types of eye colors here in these Aedes aegypti females. In the past, in our lectures, we've discussed Mendel's work. Things were pretty black and white for Mendel. Genetics is a little more complicated than just dominant and recessive alleles. In this lecture, we are going to explore some of those complications. To start out with, I'd like to talk about the interactions between alleles of the same gene. The classic example of interactions between alleles is complete dominance. Complete dominance is an example of a very simple relationship between two alleles of the same gene. A dominant allele and a recessive allele. And using this, Mendel was able to establish his first law that two alleles will separate at equal probability. In the parental generation, if you cross a homozygous round pea plant with a homozygous wrinkled pea plant, you will get all round progeny that are heterozygous. So R was dominant, big R was dominant, and R masked the recessive allele, which is wrinkled. If you cross these F1s in the F2 generation, you will get mostly round peas and then some wrinkled. And the phenotypic ratio is 3:1, 3 round to 1 wrinkled. And the genotypic ratio is 1/4 dominant, 1/2 heterozygote, and 1/4 recessive. Alleles of the same gene do not always interact in this way. You can have incomplete dominance. The eggplant here is an example of this. So you have a purple eggplant and a white eggplant. The purple eggplant carries 2 copies of the purple allele, and the white eggplant is homozygous for the white allele of the same gene. If you cross these in the F1 generation, you get something in between. You get a violet eggplant. So incomplete dominance is exhibited when the heterozygote has an intermediate phenotype. So if you cross these two F1s together, in the F2 generation, you do your Punnett Square, you will get a genotypic ratio of 1 to 2 to 1, and a phenotypic ratio of 1 to 2 to 1. So let's go back to the mosquitoes. Here, I've mutagenized these mosquitoes for an eye pigment gene. The wild type is pictured here on the left, and it's in the circle that has just come up. So that's the darkest eye pigment. A heterozygote for this gene has an intermediate phenotype where its eye is slightly lighter. And the homozygous mutant has a very light eye. Codominance is similar to incomplete dominance, but there's an important difference. The phenotype of the heterozygote includes the phenotypes of both homozygotes in codominance. But the difference between codominance and incomplete dominance is in the expression of the alleles. Codominant alleles are both expressed. Incompletely dominant alleles, only the dominant allele is expressed. So the heterozygote shows, let's say, lighter eggplant color because it has less of the gene product. So usually the recessive allele is a loss of function allele, an allele that disrupts the function of the protein. So some examples exist of this in humans that are related to human health, such as cystic fibrosis and sickle cell anemia. The cystic fibrosis gene, cystic fibrosis transmembrane conductance regulator, CFTR, has been mapped to chromosome 7. And heterozygotes express the functional protein, are healthy. Carriers, heterozygotes, express both the mutant and the functional protein, so is therefore codominant and are healthy. If you have two mutant alleles, both of the alleles will make the mutant protein, and you'll have cystic fibrosis. Sickle cell anemia is similar. In sickle cell anemia, you have different alleles of hemoglobin. You can have wild type hemoglobin and have a purely functional protein and no anemia. So you would have wild type red blood cells. You can have one copy of the sickle cell anemia gene, which would be linked by S, versus A, the allele, the wild type allele. Here you have the expression of both types of hemoglobin, and both mutant and healthy proteins are codominant. There's no anemia, but there is some changes in the red blood cells, and you can get these sickle-shaped red blood cells in low oxygen environments. So this is sort of an example of incomplete dominance. So the distinction between codominance and incomplete dominance is less than clear. So I think they're kind of related concepts. If you're homozygous mutant, both alleles make the mutant protein. You have severe anemia, and you have sickle-shaped red blood cells. Now, sickle cell anemia, beyond the genetics of it, is interesting in and of itself, and I wanted to discuss this a little bit more. So this allele that alters hemoglobin was likely selected for as a way to resist malaria infection. This is an interesting example of how disease has affected human evolution. So here's a video from HHMI on sickle cell anemia, and I hope you enjoy it.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under certain conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single code letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in the position where glutamic acid should be. The valine makes the hemoglobin molecules stick together, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
Let's return to the eggplant. If we cross two heterozygous eggplants, we would get the expected genotypic ratio of 1 to 2 to 1. But the phenotypic ratio depends on if the trait exhibits dominance. So if P is dominant, big P, you should get, let's say, 3 purple to 1 white eggplants. If P shows incomplete dominance, you should get these types of phenotypes, that is 1 purple to 2 violet to 1 white. If the two alleles show codominance, you should get these 3 types of phenotypes. 1 purple to 2 patchy, some sort of mix of white and purple, or white and violet, potentially, and 1 white. So what are the factors that influence the expected ratios? Penetrance and expressivity are important concepts in genetics. at times, the genotype does not always produce the expected phenotype. Here I'm showing polydactyly. So these are polydactyl feet, and this also happens with hands. And you have extra fingers or toes. Normally we have 5, here we have 6. Penetrance is defined as the percentage of individuals having a particular genotype that express the expected phenotype. The expected phenotype here is having extra fingers or toes. But it doesn't matter how many. It's just as long as you express some extra fingers or toes, and it doesn't matter how well formed they are or anything like that. Just a little, if you have extra fingers or toes, you are affected. So let's say 42 people carry the gene, 38 have the condition to some degree. The penetrance then is 90%. So it's a very strongly penetrant phenotype. However, there's variability in the expression of this phenotype. Some individuals have an extra finger or 2, maybe. And this is an example of variable expression. And this variable expressivity is due to other factors, genes and or the environment, or things that we have not discovered yet. Expressivity is kind of the strength of the phenotype in an individual, the degree to which a trait is expressed in a given individual. And for example, if all people who were affected by this polydactyl condition only had 6 fingers and 6 toes on both feet and both hands, the expressivity of the trait would be very strong and not very variable. There are also alleles that are lethal. So consider the yellow locus in mice. So you have 2 heterozygotes for this yellow allele. And this was discovered by Lucien Cuneau. So the expected phenotypic and genotypic ratio, when you do the Punnett Square, are 3 yellow to 1 white. And the genotypic ratio is 1 to 2 to 1. Now, what he observes is there is actually a 2 to 1 phenotypic ratio. This is a peculiar pattern of inheritance, what he observed. 2/3 were yellow and 1/3 were white. So why was this? All the yellow were heterozygous. So he concluded that if you've got 2 copies of the dominant allele, that it must be lethal. So these lethal alleles modify Mendelian ratios, obviously. So you would get in the F1 generation, a quarter of the progeny would die. So the yellow phenotype is dominant. The lethal allele is recessive, as it is only lethal when homozygous. Now you can have more than just 2 alleles. But you can only carry 2 alleles at a time because we're diploids. So multiple alleles exist in a population, and they are known as an allelic series, and they contribute to a particular phenotype. For example, there are 3 alleles for duck feather patterns: mallard, restricted, and dusky. So if you get 2 copies of mallard, your phenotype is mallard. If you get mallard and dusky, mallard is dominant over the dusky phenotype, so you get a mallard look. If you get mallard and restricted, you get the restricted phenotype. And if you get 2 copies of the restricted, of course, you would get a restricted phenotype. And if you get 2 copies of dusky, then the duck looks dusky. Let's look at the interaction between alleles in a little more detail. So let's cross a restricted and a mallard of these genotypes, so we can look at all the 3 alleles and how they interact with each other. So we should produce a variety of gametes here that would produce 4 types of genotypes, right? These genotypes will lead to these phenotypes. You get 1/2 restricted, 1/4 mallard, and 1/4 dusky. From this, we can think about all the different genotypes and phenotypes and figure out which alleles are dominant to each other. So restricted is dominant over mallard, which is dominant over dusky. So restricted is the most dominant allele. Let's consider another example of multiple alleles, the human ABO blood groups. So in this case, we have 4 different phenotypes, A, B, AB, and O, caused by 4 different genotypes, caused by 6 different genotypes. You can either be AA or AO, which is denoted by the little I, BB or BO, AB or OO. If you are A, you express antigen type A, and the antibodies made by your body are against group B. If you express the B blood type, you make the B antigen, and the antibodies made by the body are against A. If you are of the AB blood type group, then you express both antigens A and B, and the antibodies made by your body don't exist for either A or B. So you have no antibodies to A or B. If you are blood type O, you don't produce either of the A or B antigens, but you make antibodies to both A and B. Here you can see blood recipient reactions to donor blood antibodies. So the universal donor, O, is in red. So you can see with the universal donor, the O blood type, that the recipient doesn't react to the donor blood. However, if you look at the universal recipient, which is in blue, you can see that the universal recipient can get blood of any type. The recipient will not react. Now there are other interactions that cause changes in phenotypes, and these are interactions between multiple genes. Gene interactions can produce novel phenotypes. For example, in peppers, you have 2 different genes that control the color of the pepper. If you cross a red pepper with a green pepper, you get a heterozygous red pepper in the F1 generation. And you can get, if you cross this F1, peppers in 4 different colors. So here is the Punnett Square for this dihybrid cross. And you can get a number of genotypes. If you do this F1 cross, you will end up in the F2 generation getting 9/16 red peppers, 3/16 brown peppers, 3/16 yellow peppers, and 1/16 green peppers. So you can see here on the left is the Punnett square drawn again with the genotypes highlighted that connect to the phenotype. Gene interactions don't happen only in plants, they also happen in animals. And one example is the chicken's comb. So here we have 4 different types of chickens, and their comb phenotype is determined by genes at 2 loci, at the R loci and at the P loci. So this creates 4 possible phenotypic combinations that are driven by these genotypes. So when there is the spacer, that little underline there, that allele could be either dominant or recessive in that position. So we have the walnut comb, which is determined by the presence of the 2 dominant alleles. The rose comb, which is dependent upon having a dominant R gene and being homozygous recessive for the P gene. If you have a P comb, you're homozygous recessive for the R locus, and you carry a dominant allele of the P locus. And if you have, I think the nicest looking one, the single comb, you are actually recessive for both genes. This brings up the subject of epistasis. And here, this Punnett square from Wikipedia is actually, I think, pretty cool. And it shows a kind of pretty simple idea. If you have a gene for blonde hair or a gene for red hair, right? But you have a gene also for baldness, if you add that in, when you're in your 30s or 40s, or I don't know, perhaps in your 20s, It doesn't matter what kind of hair color you have because you're going to have no hair because you're going to be bald. So epistasis is a genetic interaction between genes when one gene modifies the effect of another. For example, if gene A masks gene B, then gene A is called the epistatic gene, and gene B is called a hypostatic gene. Epistatic genes could be dominant or recessive in their effects. And now I'm going to go through these in a little more detail. Dominant epistasis can occur, and here we're showing some summer squash. There are 3 colors of summer squash that you can get: yellow, white, or green. So there are 2 loci here in this dominant epistasis experiment. The white loci, which is a W, and the yellow loci, which is Y. In the P generation, if you take a homozygous squash for both genes, which is white, and you cross that with a green squash, which is recessive at both loci, in the F1 generation, you'll get all white squash. So if you cross these squash, you would get an expected ratio of 9 to 3 to 3 to 1. And this is a phenotypic ratio. But the actual phenotypic ratio you get is you get 12 out of 16 squash that are white, 3 out of 16 squash that are yellow, and 1 out of 16 squash that are green. So how does this happen? So this is what you get. So you would expect from this cross that 9 out of 16 would be carry the white dominant and the yellow dominant allele. And then 3 out of 16 would carry the white dominant allele and be recessive at the yellow locus. You would also expect 3 out of 16 to be recessive at the white allele and then carry a dominant yellow allele. And 1 out of 16 would be recessive at both loci. But what we observe is something very different than the expected ratio. When we have a dominant white allele and a dominant yellow allele in the same squash, we end up with a white squash, not a yellow squash. When we have a dominant white allele and recessive alleles of yellow, we end up with a white squash, which is expected. If we have recessive white alleles and a yellow, then we get a yellow squash, which is expected. If you're homozygous recessive at both loci, you end up with a green squash. So what you end up with is 12 out of 16 white, 3 out of 16 yellow, and 1 out of 16 green, just as shown on the left. So let's back up a bit and look at the Punnett square. You can see here that white masks yellow, and this includes the dominant version of the allele. It also obviously masks the recessive version of yellow. So this is dominant epistasis. So how does this particular dominant epistasis work? Here we have an enzymatic pathway that converts compound A into compound B, which produces a green color, and then compound B is converted into compound C, which is a yellow color, by enzyme 2. Plants that are homozygous for the recessive white allele produce enzyme 1. This converts compound A, which is colorless, into compound B, which is green. The dominant allele at the white locus inhibits the conversion of A into B. So therefore, if you have the dominant allele at the white locus, this enzymatic pathway is blocked from proceeding. Plants with the dominant yellow gene produce enzyme 2, which converts compound B into compound C, producing yellow squash. If you're homozygous recessive for the yellow gene, you cannot form a functional enzyme 2. So therefore, you can't convert compound B into compound C, and you can't make a yellow squash. So the position of the white gene in this enzymatic pathway causes it to be the dominant player in color determination in squash. What about recessive epistasis? Here we are looking at coat color in the Labrador Retriever. Uppercase B is for black pigment. It's the dominant form of the allele. Lowercase b is for brown pigment. It's a recessive form of the allele. E is another gene, and that is responsible for the deposition of the color. Little e is a form of the E locus that prevents the deposition of color. So the coat will be yellow. So if you look at these genes, B encodes black pigment, E encodes color deposition. So if you have the dominant forms of these genes, you'll have a strongly pigmented coat if you're a Labrador Retriever. If you're homozygous recessive at the B locus, the coat color will be brown. And if you're homozygous recessive at the E locus, you won't deposit much color. So you'll be more yellow. So you would expect from this, again, the 9:3 to 3:1 ratio. But you observed that 9 out of 16 were black, 4 out of 16 were yellow, and 3 out of 16 were brown. Looking at the genotypes of these, you see that if you carry both dominant alleles, the dog has a black coat color. If you're recessive for the yellow locus, then you don't deposit color well. So it doesn't matter what pigment gene you have, the coat color is yellow. If you're homozygous recessive at the B locus, then you're brown, but only if you have a dominant allele of the E color deposition gene. So E mass B in black pigment production, because it's not deposited. E mass little b, the brown pigment production, and this is recessive epistasis. Unless a dominant allele of E is present, you can't detect the phenotype of the dominant or the recessive pigment genes. Therefore, E is epistatic to both alleles of the pigment locus B. Complementation testing is a tool that is used in genetics all the time. A complementation test determines whether two mutations occur at the same locus by crossing them with each other. Say you have a mutation A and B. If you cross and you have homozygous lines of A and B, you cross them together. If you get the mutant phenotype, then they are the same gene. If A and B are not in the same gene, then you would get the wild type phenotype. And this means that they complement each other. So genes that are alleles of the same gene, they don't complement each other. You still get the mutant phenotype. One thing to remember about complementation testing is that it only works for recessive phenotypes. So from the first part of this lecture, here are some genetic terms that I think you should know. Complete dominance. A heterozygote has the phenotype of the dominant allele. In incomplete dominance, a heterozygote has an intermediate phenotype between the two alleles, and this is the example of the eggplants. In codominance, the heterozygote simultaneously expresses the phenotypes of both alleles. Penetrance is the percentage of individuals of a genotype that express the associated phenotype. Expressivity, the degree to which a trait is expressed. Epistasis, the genetic interaction of two independent traits. usually where one gene masks the effect of another gene. The epistatic gene masks the effect of the hypostatic gene. A complementation test determines whether two mutations occur at the same locus. Come back in a bit and listen to the second part of the lecture. Now we're going to return to the subject of sex and heredity. If you remember from the last lecture, the Y chromosome is very important in sex determination in mammals. There are 350 genes. The male sex chromosome contains the SRY gene, which is critical in inducing male characteristics. And the absence of the Y gene is required for female characteristics to develop in mammals. So what are sex-influence characteristics? This is when expression of autosomal genes is influenced by the genetic sex. These genes are inherited in Mendelian fashion, but the phenotype depends on the sex of the individual. An example is the beard in goats, as you can see on the right. There is a gene that determines whether goats have this beard or not, but it is dominant in males and recessive in females. If you have the bearded allele and you're homozygous for it, both the male and the female will have a beard. However, if you're heterozygous, only males will be bearded. The female will be beardless. Of course, if you have two copies of the beardless gene, which is denoted by B+, you won't have a beard, whether you're a male or a female. So if we cross a beardless male, B+, with a bearded female who is homozygous for the beard-inducing allele, when we cross these, we should get heterozygotes that should both be bearded, potentially, because perhaps the bearded locus is dominant. But no, we get a phenotype that is determined by sex. So only males become bearded and females are beardless. So let's look at the crosses here. So what I'm showing here is an F1 cross, and this time I'm including the sex chromosomes. So we have a bearded male who is heterozygous and a bearded female who is also heterozygous for this beard determining locus, this gene. So these are the gametes that he can produce, and these are the gametes that she can produce. If we do a Punnett square of these, we see that we can get these different genotypes. The female is beardless, but if she's heterozygous for the bearded allele, she'll still be beardless. So this is the influence of sex. And you can see this here only when the female gets two copies of the beard-inducing allele does she become bearded. So for the female, you get 3 beardless goats for every 1 bearded goat. For the male, if you inherit a bearded allele, then you're going to be bearded. So you get the opposite phenotypic ratio. So you get 1 beardless male for every 3 bearded males. So sex causes clear differences in heterozygous individuals. Sex-limited characteristics are an extreme form of sex-influence characteristics. These are autosomal genes that can only be expressed in one sex. In the other sex, there is zero penetrance of the phenotype. Examples are feathering in chickens. So you have a cock-feathered male that is homozygous for the cock-feathered allele. Females can be of any genotype at this locus, and they do not express cock-feathering. For this locus, males need to be homozygous for the recessive allele, in order to show cock feathering. Another example of a sex-limited characteristic is precocious puberty in humans. Precocious puberty is male-limited. Here we have a picture of a boy, Eric, who has been affected with this condition. Usually, people affected by this condition undergo puberty at an early age, less than 4 years old. And this is a very rare condition. The secondary sex characteristics develop. They become fully fertile males at this early age. And they are shorter in stature since their long bones stop growing. It is caused by an autosomal dominant allele, P, which is expressed only in males. And usually affected males are heterozygous. If a male that has undergone precocious puberty mates with a female that has undergone normal puberty, and he is heterozygous for this autosomal dominant allele, in the F1 generation, half of the males will undergo precocious puberty, and none of the females will. Even if the female carries the dominant precocious puberty allele. So both males and females can transmit this sex-limited trait, but only males can express the trait. Cytoplasmic inheritance is another interesting twist on inheritance. It was first discovered in 1909 by Carl Correns. In plants, he found that it did not matter where the pollen came from, and this is only for certain plants, obviously. The seed origin determined the phenotype. Let's see what he did. He crossed flowers from white, green, and variegated plants in all combinations, both male and female. And from these crosses, he concluded that the phenotype of the progeny is determined by the phenotype of the branch from which the seed originated, not from the branch on which the pollen originated. He found that stem and leaf color exhibits cytoplasmic inheritance. So this is something that happens outside of the nucleus. So what could that be? What makes plants green? Well, what Carl Correns discovered here is that the chloroplasts are what are being inherited here. And some chloroplasts are unable to make chlorophyll, and therefore the plant tissue is white, whereas in some cases the chloroplasts are normal and you get green tissue. The variegated plant has a mix of chloroplasts that are either wild type or mutant for the chlorophyll gene. Mendelian inheritance works for chromosomal DNA in the nucleus, but it doesn't work as cleanly for the DNA that is contained in organelles that are outside the nucleus. In animals like plants, the mitochondria is an organelle with its own DNA. The human mitochondria contain 37 genes, whereas the human genome contains 21,000 genes. But those 37 genes can be important. The inheritance is much more complex since there's multiple copies of mitochondria in a cell, let's say the egg cell that helps form the zygote, which is pictured above. It is possible that there are complex combinations of wild type and mutant mitochondria containing different alleles. This could lead to a variegated effect depending on how many mitochondria of a given type are inherited. There is no known mechanism for equal distribution of mitochondria. And for example, the human disease Leber hereditary optic neuropathy, which causes a loss of vision, is a maternal effect disease caused by a defect in mitochondrial genes. Let's imagine this cell, which is dividing, is a zygote, and it contains an equal number of mitochondria with wild-type genes that are in red and mitochondria with mutated genes that are in blue. The mitochondria segregate randomly during cell division. They replicate, and you get random segregation in subsequent cell divisions. This can happen in such a manner that you can get some cells that only have wild-type mitochondria and other cells that only have mutant mitochondria. Genetic maternal effect is not limited to the inheritance of mitochondria. The phenotype of the offspring is simply determined by the genotype of the mother, and this can include the mitochondria. It is determined by substances present in the cytoplasm of an egg. The shell coiling of the snail, either dextral or sinistral, is a good example of this. If the shell coils to the right, it is known as dextral coiling. If it coils to the left, it is known as sinistral coiling. The dominant allele, S+, induces dextral coiling, and coiling depends on the mother's genotype alone. If we do a cross between a dextral male and a sinistral female, both of which are homozygous for those alleles, all the F1s are heterozygous because the genotype of the mother determines the phenotype of the offspring. All the F1s have a sinistral shell. If we look more closely at this, the mother will undergo self-fertilization, and then the F2 generation will all be dextral, even though their genotypes are not necessarily associated with being dextral. In other words, because the mother of the F2 progeny has the genotype S+S, so she carries the dominant allele, all the F2 snails are dextral. Genomic imprinting is another example of maternal effect or paternal effect. Expression of a particular gene depends on which parent it came from. In the case depicted on the left, the size of the embryo is controlled by a gene, LGF2. The LGF2 allele from the male parent is expressed. The LGF2 allele from the female is silent. Methylation of LGF2 DNA is responsible for this difference in expression. And this is an example of epigenetics. Epigenetics can have a profound effect on a gene's expression. Epigenetic effects are phenotypes that are passed to other cells and sometimes future generations that are not the result of the change in DNA sequence. Some examples depicted here are histone methylation, histone acetylation, and DNA methylation. And these are all epigenetic marks that can alter the chromosome and change the expression of a given gene. Even though these marks can be inherited, they can also be removed. So the effect of these epigenetic marks is not as permanent as changes in DNA sequence. And we can see this very clearly in anticipation. In anticipation, the onset of a disease can vary as it is passed from one generation to another, appearing earlier than the previous generation. Huntington's disease is a clear example of this. The disease is caused by a gene on human chromosome 4, which is depicted here. This gene contains an unstable region of DNA, and this unstable region can expand and contract. The degree of expansion is proportional to the severity of the disease. Rapid expansion is seen when the gene is inherited from father to offspring. So here we have a figure showing these triplet repeats that can occur in the Huntington's disease gene. So a healthy gene only has a few repeats, whereas a disease gene has many more. So the disease is caused by this unstable region of DNA that increases in size from generation to generation. Now we're going to turn to some other important genetic concepts: pleiotropy, polygenic traits, and continuous traits. Here is a man with Waardenburg Syndrome. You can notice that his eye colors are different. One eye is blue and the other is green. There's also other changes in the coloration of his hair. This is caused by a pleiotropic gene. A pleiotropic gene, multiple, often seemingly unrelated physical effects, are caused by a single altered gene or pair of altered genes. Waardenburg Syndrome is a very clear example of this. So you get varying degrees of hearing loss and changes in skin and hair pigmentation, as is shown above. The syndrome got its name from a Dutch eye doctor named Petrus Johannes Waardenburg, who first noticed that people with differently colored eyes often had a hearing impairment. Mutations in multiple genes can cause this syndrome. These genes are involved in the formation and development of several types of cells, including pigment-producing cells called melanocytes. Traits can also be polygenic. So this is the production of a single trait due to the effect of multiple genes. The color and patterning of the corn snake is a great example of this. You can get a great variety of colors and patterns in corn snakes, and this is done by breeders who are interested in generating interesting pets for their customers. Here are some examples of what the breeders have come up with. And you can really get quite a diversity in the coloration and patterning of this species through breeding. Human height shows a great deal of variation. Here is an image taken from 1914 of students at a college, and they're lined up by their heights from 4 feet 10 inches to 6 feet 2 inches. Here is an image from 1997 at the same college, which shows the distribution of heights. And if you look carefully, you see that more people are taller. And this can be due to multiple environmental and possibly genetic effects. But it seems that as a human population, we're growing. Human height is an example of a continuous characteristic. So there are discontinuous characteristics that have distinct phenotypes. Others exhibit a continuous distribution, similar to the graph shown at the right. An example is the heights of individuals. This is controlled by many genes, so it's a polygenic trait. And this is called continuous characteristics. You can describe these characteristics using quantitative terms. And these are quantitative traits. Quantitative traits are characterized by continuously distributed phenotypes. Differences observed between individuals for quantitative traits are thought to reflect differences at numerous genes. These are also referred to as polygenes or quantitative trait loci, QTLs. So if we have one loci with two alleles, big A and little a, the number of genotypes we would expect are 3. If we have two loci with two alleles, the number of genotypes we would expect are 9. Continuous characteristics can be influenced by non-genetic determinants such as environment. And they are multifactorial as many factors influence the outcome. The environment can have a profound effect on how genes express their phenotypes. In this case, the range of a phenotype is determined by the environment. And there is a great example of this in the Himalayan allele in rabbit. Rabbits that are reared at 20 degrees C or less have a different pigmentation than rabbits that are reared at temperatures above 30 degrees Celsius. This is because the enzyme that produces the pigment is inactivated at high temperatures. This is therefore a temperature-sensitive allele, which produces a range of phenotypes based on the temperature. So we've covered a lot of ground today, and I want to leave you with some genetic terms for you to remember. Sex-influenced traits are encoded by autosomal genes that are more readily expressed in one sex. Cytoplasmic inheritance, a trait encoded by genes located in the cytoplasm. Genomic imprinting, genes whose expression is affected by the sex of the transmitting parent. Epigenetic effects are phenotypes that are passed to other cells and sometimes future generations that are not the result of changes in DNA sequence. Anticipation is the stronger or earlier expression of a genetic trait in subsequent generations. Polygenic traits are encoded by genes at many loci. Pleiotropy, one gene affects multiple characteristics. Well, I hope you have enjoyed today's lecture. Here's a little preview of our next lecture, which will be about pedigree analysis for the most part. Here is the inheritance of hemophilia in royal families in Europe. See you next time.